Hello, everybody, and welcome to our final day. I know it's been quite a meeting. Some of us are getting used to doing these all-day meetings again after COVID and virtual meetings. These all-day in-person meetings are still a little bit different than some of the virtual ones, but I think great to have and really great energy. And for these reflections, we all sat and sort of thought about the things we've seen yesterday, but also over the course of the meeting. And there are really two things that stood out to me. And one was really, the desire and the push for harmonization and greater harmonization across the exposomic space. Yesterday, we had a couple of really great talks that highlighted just the sheer number of cohorts that are, working in exposomics across the world for it, many of which I had never heard of, certainly, and I think many in the audience certainly had not heard of either for it. And if we're going to utilize all of these resources, these cohorts, we all recognize the need for harmonization, the need for a shared language amongst them, and the need for shared standards and approaches and ways to translate across these different databases. And I think that's something that the field's going to develop as we go about this moonshot effort. And I think the second thing that I sort of took back myself from the past couple of days is really the strong desire for a shared vision amongst the exposomics community. There were several, talks I had where people were, asking, okay, well, what's going to be the, organizing body for this? What's going to be the overarching sort of vision, the overarching goal that's there? We have the moonshot in there, but, what are we going to consider to be our moon? And what's going to be the next launching pad after we, get through the moonshot, hopefully in the next, 10, 15 years? And so I think, developing, workshops like this and developing that shared vision, that shared purpose, that shared goal, and then not just having one launch, but multiple launches for it. Perhaps an international space station, will be sort of the next thing after we complete the moonshot for it. I think it's something that the field really is looking for and is really building right now.
Good morning. First of all, I want to thank all the members of the organizing committee actually for the invitation. It's been a pleasure to meet all the experts in the field. And it's been really amazing to see, and I agree with Cavin on the number of cohorts. So we know that when we're conducting research, we see the publications, we see that we have different birth cohorts or even cohorts in general. I mentioned the birth cohort because we were working on a birth cohort, but different cohorts were mentioned that we, at least on a personal level, I don't know about, and it was really amazing to see the work and the results. And I wanted to actually add on what Cavin was saying, that we really need harmonization. And one of the points that actually I really noticed and I want to add on it is that we need this global collaboration, but at the same point, we have at the same time to think about the customized approach, because we have, like in the toxicology, I don't know if Fiona is here or not, but going back to the individual biological variation, we have to think also about more of country-specific or region-specific variations, and this leads us to have a kind of milestones adapted to different contexts, even though we have this global approach and this global support, and it's really amazing to see how people are ready to help and ready to support each other. And by having these open data and open collaboration, but it's really important also to take into consideration that some regions or some initiatives are not yet complete and need this customized approach. So we were supposed to reflect a bit also on our commitments, and from my side, and I guess it can be shared and we can see the audience later on their input, I see that one of the most important commitment that we need to take after this meeting is to have this inclusive actually research approach or design, taking into consideration the difference between different countries, having this long term to commit to have this long-term collaboration, and to commit to actually transfer it to our countries, because as I said, we have different specificities, and taking Lebanon as an example, we have to work on it differently. It took us, for example, 4 years to approve the roadmap for climate change, so we need to follow a specific process. The third one, I would say, it's more about ethics, and it was mentioned also during the sessions, ethics and data management, but at the same time more all of regulatory actions. And the last point, which is very important actually for me, and I guess you will agree maybe, the public health awareness, but at the same time, the capacity building, and here involving community, and this to add to what has been said since day one. So we cannot move or we cannot act in different countries if we do not have all the stakeholder support, whether the government, communities, NGOs, and academia. So this is actually my input, and thanks again for this great opportunity.
Good morning, everyone. My name is Peng. First of all, I just want to thank the organizing committee for having me on this panel, and it's a great pleasure to attend this meeting as well. Really nice to meet all the old friends that we met on at the Banbury conference as well, and really feel it's like a large version of it and generating more transformative and this interdisciplinary idea to trying to push this field forward. There are some thoughts, my personal thought I want to share. The first thing is, and I think during the discussion yesterday, I feels like usually we are underestimated how complex and how heterogeneous the exposome is compared with the human genome, because exposome is a really personalized cloud. Definitely, genome is also personal, but it just have the ATGC, and based on sequencing, we can achieve it, and during the lifetime, it doesn't change, right? But based on what we found when I was a postdoc at Stanford working with Dr. Michael Snyder, and then the current group at Harvard School of Public Health, we already have a lot of evidence showing that the individual levels exposome keep changing, depends on the location and time, and they are super diverse and dynamic. Also, we are exposed to thousands of the organic, dozens of inorganic, millions of microbial, and our mental status is also keep changing. So it's a really complex interaction between different parameters. That's the reason we want to feature the data harmonization. Also, is super high dimensional compared with the genome data set. So that means that we really need input from different fields rather than just focus on toxicology, exposure science, epidemiology, engineering, etc., isolated or separately. I really feels like the future collaboration is necessary for this field, and also can generate a more high impact research or outcome in this field. And yeah, last but not least, just a personal note that I think we really need to increase some visibility of the term of exposome or exposome mix in the biomedical science or other fields, because every time I attend some other conference, people will ask me, what are you working? I will say exposome. And the next question would be, what is exposome like? Can you briefly introduce? So yeah, I think everyone here is the ambassador of the field. So I highly recommend everyone to talk and introduce this to all the potential reviewers, or your colleagues, funding agency, government agency, etc. I think that would be really helpful. Yeah, thank you.
Yeah, thank you very much also to the organizers for having me, and it's been a great few days. It's hard to add something on what Cavin, Myriam, and Peng already said, but I'll just try to reflect a little bit what I felt. As you now all know, I'm a toxicologist after last night. So I see it through my glasses, and I'm also coming more from the environmental field, so I'm a bit new kid on the block of exposomics, but I find it really interesting. It's nice to start a new career at the end of your professional life. So my takeaway from the session, global coordination, collaboration last morning, the research infrastructure as models, was really that Nexus and all those other infrastructures we've heard of are amazingly advanced and super open to collaboration. So Robert Moulin-Barouki, sitting up here, pointed out that he is used to that will get a stick on you. Is pointed out that he used to he's used a competition between the leaders in the field. I didn't cite him exactly, but I just noted that down because I felt it was important. But when it comes to exosomics, he said he felt the willingness to collaborate and be completely integrative or something along those lines. And I can only say yes to this. This is also how I perceive the field. So these initiatives are really eager to develop a common language and are eager to move from discovery to intervention research, but they also need to grow together. I thought I'm an outsider as a toxicologist, but no, we have engineers, we have geologists, we have everything here. And so I think we still need to move more together and learn a common language, but I think, yeah, these 3 days are a really good first start. Then I found the group sessions amazingly productive. So I had not expected that this 1-2-4-all approach works so well, but it worked really well also because everybody was so super motivated, and that a group so new and from so different ways of life. I mean, I literally didn't know anybody in my group before, but now I feel like I've made a lot of friends. They were just, discussing productively. I was in a group of big data and AI, which is certainly not my field because I'm still in they call it your wet lab. You know, my lab is robotics. It's not wet or so. I mean, it's a little bit wet, but cells need water. We all need water. But so this distinction, I think, is pretty smooth between computational and experimental. So there's a lot of computational in experimental, and we have to recognize that as a byline. But in this group, I think we did a real good job going from low-hanging fruits to get us going by exploiting existing data, so not being paralyzed, what often happens in different fields, that you say, oh, it's too difficult, I don't even start, but there's this willingness, yes, we have only 20 chemicals, but let's get started. We know there's some 200,000, but let's just get going, and then develop a long-term goal to create a human reference cohort, potentially even a virtual cohort. We talked a lot about digital twins, which I know really well. I'm in the field Earth and Environment at Helmholtz, and so they're all doing digital twins, and I thought I can never do that in toxicology, but I'm going back, and I think that's, I have a better understanding of that. And I must admit that I saw the potential of a deeper understanding of the exposome, mainly for public health, before I came here. And possibly also improvement of the environment, but I learned about this idea of a personalized exposome that you all mentioned. That wasn't that clear to me, that helps each individual to manage their own health, which I found very appealing. So we were even going to this, that you get your own exposome later. But then thinking about that later in the night, same thing, so I didn't sleep all that well, despite I thought I was exhausted. So it occurred to me that we still have a very anthropogenic view, in that by understanding and managing the exposome, we can improve human health. That's kind of our paradigm. And so I think what we neglected then in the whole discussion was the impact this work can have on improving our environment. So if we identify lots of plastic chemicals and associate them to the onset of metabolic diseases, which we're almost there in this direction, that would give us more authority to phase out plastics. If we see that a lack of green urban spaces has a negative impact on the exposome, we have a mandate to improve our urban environment. And I think that's maybe the next step, but that's something we haven't really discussed here, so I just wanted to bring that up. And so we also started yesterday to become much more concrete than on day one, but I still feel that our discussion is very hypothetical and theoretical, and we are sometimes also lacking some of the pragmatism and focus on the practicability. So I was not in the group that put the kit or the chip forward, but I would warn and to be reconsidering it a little bit, because we have it to do with nasty chemicals, and the problem of nasty chemicals is the nasty ones are toxic. The ones that stick to plastic and they stick to everywhere, get lost, don't operate well, and so I think there's a real problem that we maybe have to dream not too far, but make sure that we really encompass the entire chemical space. I have much more, but and especially on I didn't get to toxicology, but I give up. I said a lot about that yesterday. Yeah.
I love the enthusiasm and the passion and the excitement. It's fabulous. The reason I stepped up is just because I wanted to make sure we had an opportunity to hear from a couple people from the audience. What are you reflecting on this morning that you'd like to add to this conversation? Please go ahead.
Hi, my name is Victor Bornstein, and I wanted to bring light to a topic that I don't think was discussed that much yet, but for me is very important. Sure. And when it comes to the final goal of us really creating a better environment, creating better health, we discuss policymakers. And I wanted to highlight a different ally that I represent, which is the litigation community. Like for PFAS litigation and EPA work, it was super crucial, the work the litigators did. So I just wanted to highlight that as something like bringing actionable insights and change in society quickly, because litigation is faster often than policymaking. So if anyone believes that their discovery, their compound is ready for being acted upon legally, if they can come talk to me, I would be very happy to talk about that.
Wonderful invitation.
Thank you.
And that's what this conference is all about. That's what we're here for, is making connections. So thank you for making that invitation.
Yeah, thank you.
Fabulous. Yeah.
There it goes. So I'm Ben Place from NIST. And so one of the things that I've been kind of sneaking around and talking about, but we talk about harmonization, standardization, is in something that hasn't come up in the bigger groups, are the National Metrology Institutes. So NIST is the US National Metrology Institute, but we are partners with National Metrology Institutes all across the world, who are the people who develop the reference materials, the people who do inter-laboratory studies. Some do proficiency testing. If we're talking about harmonization, if we're even talking about validation of chip techniques, a lot of the things that we can do is when we center ourselves on standards, on reference materials, it allows us to improve comparability and really be able to ground ourselves in something like the SI. And so I don't really have the answer, and I think that one of the things that I was hoping to get out, and I do have I have connections now, people to talk to, is how the NMIs can serve the exposomes community. We can make reference materials, but we need to make sure that they're fit for purpose and they answer the questions you need. We had a big conversation last night about some of the reference materials, but they need to have things in them to be interesting, or else it's just a standard serum or something like that. And so, please reach out to your own NMIs, especially if you're in different countries. If you don't know who your NMI is, let me know. We meet once a year, twice a year. And one of the big things that's on our head is the non-targeted, is how NMIs can support non-targeted, which includes uncertainty assessments, confidence, probabilities, all of these things that are attached to identification, so that we can kind of ground ourselves, which again improves comparability, but also kind of ground truths us. So, just heard about litigation. Litigation needs that ground truth a lot of the times. And so, if there's a way we can support those kind of actions, we just need to know what. I've been listening, and I do plenty of non-targeted, but at the same time, I need to know what would actually serve the community. So, yeah, please consider your NMIs, your National Metrology Institutes.
Fabulous. Thank you. Is that Raja? Go ahead.
Hi, I'm Arkat Rajasekhar from UNC Chapel Hill. When I looked at the whole 2 days we had great time. I enjoyed it a lot. One thing I noticed is that we were more concerned about the human aspect, the human health, which exposome can help. But I think that there is a lot more than humans on this planet, and we need to look at how other things like animals and plants are affected by exposure and how we can help them mitigate some other things. And we can learn a lot from them. For example, we know that there are certain creatures which are in the depth of the sea, where in very harsh conditions they're able to survive. What is the exposure? What is the type of things they are able to overcome and live in that particular environment? And learning that is probably one of the tools which exposome can be used to help find out what is happening there. I think looking at the larger biota is an important part, I think. Thank you.
Thank you, Raja. I think Raja just added like 15 more concentric circles. So thank you. Let me go up here.
Hello, Jennifer Kerwin, and here I have my MQAC hat on. For those of you who have not heard of MQAC, it's the Metabolomics Quality Assurance and Quality Control Consortium. I'm a former chair of the coordinating committee. I just wanted to take up the baton on reference materials and say that MQAC has done a lot for metabolomics in best practices and defining reporting standards, and has worked with NIST and other metrology institutes for reference materials. And I'd be very happy to act as a central point if people are interested in setting up an exposomics working group within MQAC and make use of the infrastructure there.
Thank you, Jennifer. And before you run away from the microphone, can you say the letters? Because if I were to try to spell NQUAC, it might not come out the right way.
Okay, so it's Metabolomics Quality Assurance and Quality Control. So it's QAQC. Consortium.
Ah, QAQC. That's easy. Okay. Thank you very much. You can go over here.
Well, this is more of a structural thing because I was in the green group. So we talked a lot about sharing. And we talk about sharing, there's also some subtext to it, which is we're talking about concept of ownership, access, and priority. If this is going to be a global enterprise that we're going to do, and like any moonshot is going to lead to new technologies that we probably haven't conceptualized yet, that's going to trigger a tech transfer problem. So we need to think ahead of time. What is the rules of ownership for the international consortium we're trying to put together? Is this going to be something that can be monetized or not? And what are the rules for us to do that so that in doing so, we don't lose a trust as being seen as profiteering purpose?
Yeah, thank you for that. That word trust is a big, big word. I think, you know, that's going to be really pivotal to success in this arena. Purnima?
Thank you. Purnima Prabhakaran, I'm from India. I just want to start by saying thank you so much for having me here. This opportunity has just been fantastic for me. I feel a sense of privilege being probably one of two people who's flown all the way from India to be a part of this conversation. And I just want to share a couple of thoughts that I've had, being here for 2.5 days now. And I was talking to Fena and others earlier in the organizing committee. And I think there has been an effort to kind of include people from other parts of the world because I was getting a sense of, there's excellent vanguards here in the form of Irene and Nexus. And I think there's lessons to be learned for people in other parts of the world. It was beginning to feel sometimes a little bit like 1090 problem. That's like an arbitrary figure that I'm taking, like only 10% of people discussing, where 90% of the burden is really. And I think we'll miss big parts of the puzzle because, not least because we're taking a one-size-fits-all approach sometimes, and we're talking of data analytics and big data and things, but I think there are problems in some parts of the world where there isn't even data. So where do we start from? So we all have different people at different starting points, and I think we need to address those gaps in a big way. Otherwise, taking something like Irene and Nexus to a global level will miss certain actors and certain pieces. And I think just taking the example of data, like we might have data in some parts of the world, and you saw the fantastic last session we had at the end of the first day. It was a whistle-stop tour of different parts of the world, and people have said time and again they were not aware of these resources. So I think one of the near-time goals should be really to do that mapping. I said that in the breakout rooms, and people said, oh, we've been talking exposomes for 15 years, but if we've talked 15 years and not had people in this room who needed to be here, then I think we had already missed the bus. So, because there is data, where there is data, is it available? Is it accessible? And when it's accessible, is it usable? And I work in a space where I spend sometimes 18 months trying to access data in my own country. So there's regulatory compliances. There's lots of roadblocks that can come in front of you. So I think it's important to have people in the conversation. And I know the effort was there. It's just that people didn't make it for different reasons. But I just want to leave with this thought.
Thank you so much. And thank you for, I think, also mentioning the roadblocks. There are roadblocks. So how can we get rid of them, right? Thank you. Gary.
Gary Miller. So following up on Raja's comment, I was at a meeting in Strasbourg, France earlier this year on the exposome and One Health. And it was very enlightening for me because people were talking about plant systems and reeds and like filtering water and things. And I think the analogy is that there's like this Earth biome project trying to do DNA sequencing on every species in the world, is that once we get exposomics working, you can use it on the ecosystem, on the plants, and all the different species. And you think about food quality or contamination in our soils, that's what we want. We want exposomics going across. But we have to start somewhere. And I think the idea of getting the techniques and technologies, I think human and models that like that first is a great starting point. But I want to see exposomics like genomics in all of biology.
Thank you, Gary. Okay, so it looks like we've sort of come to a natural close to this session. So great round of applause.